Friends, welcome to Biology Made Easy. Hope you are all perfect and we are discussing the chapter genetics and in this chapter, today in this video, we will discuss laws of inheritance. The laws of inheritance is also popularly known as Mendelism. So actually when Mendel conducted his experiment, he didn't put forth any laws. He put forth certain formulations, certain generalizations. And in 1901, Correns raised these formulations to the laws of inheritance. So it was Correns 1901, he elevated the formulations or generalizations of Mendel to laws. So there are three laws of inheritance. One is law of dominance, law of segregation, and a law of independent assortment. One is a law of dominance. The second is a law of segregation. And the third is law of independent assortment. Out of these three laws, the first two laws, they are coming under inheritance of one gene. Inheritance of one gene, and it is based on monohybrid cross. While the third law, law of independent assortment, it is based on inheritance of two genes, and it is based on dihybrid cross. So let us discuss one after another. Let us discuss first this law of dominance. So law of dominance, it is based on the monohybrid cross. So in one of the monohybrid crosses, Mendel took a pure tall plant and crossed it with a pure dwarf plant. That means it is given in the symbol T for tall, while for pure dwarf it is written in lowercase t. It is never written as D, never written as D for dwarf, because the dominant character is always in capital letter and the dwarf character is always written in this lower form. It is not written like this. So the pure tall is capital T and pure dwarf is small t. When a cross was made between this, now this will produce gametes. They are equal T form. This also produce gametes, one type, but T and T. When they will combine together, the F1 hybrid, now it became tall. And again, when this tall plant of the F1 hybrid was intercrossed for the F2 generation, it gave rise to plants. One is in the form of capital T. This is homozygous tall. Then one is capital T small t and another is capital T small t. These are heterozygous talls. And one is small t. This is homozygous dwarf. That means this is the F2. In the F2 generation, the tall is to dwarf plants. They are produced in the ratio of 3:1. This is the phenotypic ratio. There are 3 tall plants and 1 dwarf plant is there. But genotypically, one is capital T, that is homozygous tall. Then 2 are the heterozygous talls, and one is the homozygous dwarf. They are in the proportion of 1:2:1. So this is the phenotypic ratio, this is genotypic ratio. Now when we will discuss this law of dominance, what is the meaning of this law of dominance? That means each character, suppose this is tall character, this is dwarf character. Each character is represented by a factor according to Mendel. Each character is represented by a factor, and the factor of Mendel is nowadays known as gene. Then each factor has two alleles. Two alleles which are present in pairs. Now, both these pairs, that means the capital T and small t, they are present in this F1 hybrid. In the parent, in one case there is only capital T, in the other case there is only dwarf or the small t. But in the F1 hybrid, one is capital T, the other is small t. But this plant is tall. That means when the plant is in heterozygous condition, only one allele is expressing itself while other is unexpressed. Or we can tell that one allele is masking the expression of other character. So the character which is dominating, the character which is expressing itself in the F1 heterozygous hybrid, that is known as dominant. That is dominant, and the character which remains unexpressed, here as in case of the dwarf, that is recessive. So this is known as law of dominance. That means when both the alleles are present together in the heterozygous hybrid, only one will express itself, not the other one. The one which will express itself is dominant, and the one which cannot express in the F1 hybrid, that is recessive. This is law of dominance. But to this law of dominance, there are certain exceptions. So, exception to law of dominance. What are the exceptions? It is again very important for example point of view. One is incomplete dominance. Incomplete dominance. So, in case of incomplete dominance, none of the alleles is dominant over another. And the second exception is codominance. In case of codominance, both these alleles will express themselves equally, which we'll discuss in later on videos. But these two are exceptions to the law of dominance. So the first law is complete. Let us discuss the second law, law of segregation. So according to this law of segregation, when you look into this F1 hybrid, in the F1 hybrid is tall, but the genotypicality is capital T and small t. That means both the alleles are present together in the F1 hybrid, but they do not blend together, they do not mix together. That is important. The alleles do not blend, or they do not mix together. Each one keeps its identity. How? Again we see when in the F2 generation, the character which remains hidden, which remains unexpressed, is appearing here. Because when we made a cross between tall and dwarf, the F1 appeared to be tall. That means the recessive character didn't express itself. That doesn't mean that it is lost. Neither both the characters blended together. It keeps its identity so that in the F2 generation, it again appeared in the 25%. So when it will be possible? When it is possible, it is explained by the law of segregation. How? Now the heterozygous plant F1 hybrid is capital T small t, its genotype is capital T small t. When it will produce male gametes, that means the out of total male gametes, 50% will produce capital T and 50% will produce small t. They will separate and segregate. Similarly, out of the total female gametes, or you can tell eggs, then 50% will produce capital T and 50% will produce a small t. So that means each gamete is producing one of the allele, either capital T or small t. That means in case of male gametes, it is producing only capital T or small t. Similarly, in the female side, it is producing only capital T or small t. That means in none of the gametes, both the alleles are present, only one is present. That is why law of segregation is also known as law of purity of gametes. It is also known as law of purity of gametes, or law of non-mixing, or law of non-mixing of characters. Law of non-mixing of characters. That means, in this case, though in the parent both the alleles are present together, during the formation of gametes, what happens? During the formation of gametes, there is meiosis. And during this meiosis, the gametes both in the male side and female side, they separate, they segregate. So that 50% of the male gametes contain capital T, 50% of the male gametes contain small t. Similarly, coming to the female side, 50% of the eggs, they contain capital T and 50% contain small t. That is, there is separation, there is segregation of characters during formation of gametes, because at that time there is meiosis. That is why it is possible that though the both characters are present together in the F1 hybrid, they separate, they segregate at the time of meiosis during gamete formation, so that in the F2 generation, both tall and dwarf plants, and again the homozygous tall, heterozygous tall and pure dwarf plants are possible. This is known as the law of segregation. That means during meiosis, during gamete formation, the alleles, the characters, they separate or segregate from each other. So this is law of segregation. And this law of segregation, what is its importance? The law of segregation is the fundamental law of genetics, to which there is no exception. That is very important. We have discussed for the law of dominance, there is the exceptions, incomplete dominance and codominance. But for law of segregation, there is no exception. It is the fundamental principle of genetics and it is universally accepted. So it is the fundamental law and universally accepted. This is one point. The second point is, when Mendel carried out his experiments, meiosis was not discovered. But from the separation of the characters, it was predicted that meiosis is there. That means the occurrence of meiosis was predicted before it was discovered. So these are the two important points of law of segregation. Now let us discuss the third law, that is law of independent assortment, and that is also known as, and again this law of segregation is also known as first law of genetics. Now when we will discuss the law of independent assortment, it is also known as second law, and it is based on inheritance of two genes, and it is derived from dihybrid cross.